Imagine a world without the measles vaccine. Or just look around us now. We are living in the middle of a pandemic. The disease spreads quickly, resulting in huge outbreaks. And although not deadly for most people, it still causes harm, physically as well as economically. This is exactly what happens when PRSV, the porcine reproductive and respiratory syndrome virus, is introduced into a swine herd. Growing pigs show fever and pneumonia, and sows have late-term abortions. Usually, infections and vaccines stimulate the immune system immediately and eventually result in the production of antibodies. PSV somehow delays this process in pigs, making us wonder, how does the virus do that and what cells are part of it? In general, immune responses are quite complex. Imagine walking into a party, many people dancing, laughing, talking. Those people represent some of our immune cells. They're driving immune responses, or the party in this case. But what about the many people in the background? They are quiet, but not less important. They initiated and organized the party. Those people can stand for an overlooked immune cell population called gamma delta T cells. In theory, gamma delta T cells meet PRSV directly in the lung. They then influence other immune cells or even kill infected cells themselves. So far, we showed that young pigs have a high number of gamma delta T cells in the blood. It's 6 times higher than in humans. And some of them have receptors that are part of activation processes. We also showed a trend towards older pigs having more of this reactive gamma delta T cell phenotype. So they might indeed respond to external stimulation. My research will now go on to answer the questions: what happens when pig gamma delta T cells are facing the virus? And are they comparable to human gamma delta T cells? Let's assume gamma delta T cells can fight against the virus, but are not perfectly stimulated by infection or vaccination. If this holds true, it can give us a new strategy for vaccine improvement. Specifically intensify gamma delta T cell functions would be the new goal. But can this help you and me? Well, yes, it can. By discovering functional similarities between the pig and the human immune system, pigs can be an appropriate model to study human diseases and infections. So remember, every cell in our and the pig's immune system has its own purpose. Even the quiet outsiders have important functions we shouldn't ignore in vaccine development.